Prokaryotes have a profound mechanism of perception of osmotic stress. Under situations of hyperosmotic shift, when the surrounding environment has more negative water potential, water from inside of the cell tends to move out. In order to prevent this, the cell needs its own water potential to drop down to the level of the one existing outside the membranes. And for this purpose, it has to increase its own solute concentration. This solute concentration depends on accumulation of such moieties which do not disturb the ionic balance of the cell. Therefore, charged participants are generally avoided. Most popularly used solutes include the ones called compatible solutes. They can be accumulated up to very high amount without disturbing the charge concentrations or charge balance of the cell. These include trimethyl ammonium compounds that are mostly betanes, amino acids, sugars, and heterosides. Now, accumulation of these compounds can occur both ways, either by taking them up actively from outside of the cell or by synthesizing them new and fresh inside the cell, that is through de novo biosynthesis. Reverse of this has to happen during hypoosmotic shift, when the water potential outside the cell is higher as compared to that inside the cell. So under those conditions, the cell has to prevent the water from coming inside. So it needs to extrude small solutes. Now, since this process has to be very rapid in order to prevent water from coming inside, the cell does not depend on specific carriers, rather it has channels through which solutes can move out at a greater pace. A very good example of a de novo biosynthesis mechanism for accumulation of solutes includes the one for the accumulation of compatible solute named glucosyl glycerol. Now there is a transcriptional inhibitor named GGPR which does not allow active transcription from the gene for GGP synthase. So there is a basal transcription that occurs and very small amount of GGP synthase is formed. Now this protein goes and binds to the DNA and remains in inactive form. So under this condition, there is no GG synthase enzyme and no GG is formed. Under situations of high salinity, when there is excess Na plus and Cl minus in a separate series of events, an activator called SIGF binds to the same sequence where GGPR was binding earlier and promotes active transcription from GGPS gene. When there is formation of GGPS, it tries binding with the DNA, but because of excessive Na plus ions which are shielding the DNA, it is unable to bind, and therefore GGP synthase remains in active form and the transcription also continues so that more and more of GGP synthase is formed. Now since there is high amount of synthase present, more and more accumulation of GG, that is glucosyl glycerol, occurs within the cell. As a part of this mechanism, when the solute gets accumulated, water potential gets increased, no more of sodium ions are pulled inside, and the hyperosmotic stress is counteracted. Now since there are no sodium ions to shield the DNA, the GGPS that now forms goes and binds to the DNA and becomes inactive again. Therefore, the transcription shifts back to its basal levels with no more synthesis of GG synthase and no more compatible solute formation. So this loop functions in response to change in hyperosmotic condition. Another example is the kind where there is uptake of compatible solutes, here namely glycine betaine and proline betaine. For this particular gene, there is a histone-like protein called HNS which binds to the DNA and there is no transcription. Under situations of high osmolality, this HNS protein is unable to bind to the promoter region, where now RNA polymerase can bind, and there is active transcription of osmolyte uptake proteins. So in either of the two cases, the concentration or the amount of compatible solutes inside the cells rises, so that the water potential becomes more negative and the flow of water from inside of the cell to outside is prevented. Now there are several examples of membrane bound proteins which function as osmosensors as well as osmoregulators in prokaryotic systems. Some examples are Prop-E of E. coli that belong to MFS transporter family and are responsible for uptake of a range of solutes, namely proline, glycine betaine, piperolic acid, taurine, etc. Next is OPU-A from L-lactis, which belongs to ATP binding cassette transporter family, and it is mainly responsible for uptake of glycine betaine. BET-P from Corynebacterium glutamicum that belongs to BCCT transporter family and is responsible for betaine transporter. All these three act in sensing the change in osmotic potential, and they also have a capability of regulating it to some level by acting as a channel. So they may act as sympoters such as Prop-E, which helps in uptake of solutes along with protons, or BET-P, where there is symport of glycine betaine with 2 sodium ions, and likewise they try maintaining the water potential. OPU-ABC, as is visible here, is a kind of transmembrane protein that has a substrate binding subunit with it, and BET-P also is a membrane bound system where its C-terminal positively charged domain interacts with the negatively charged membrane when it is in a resting state, and when there is change in the osmotic potential, the potassium ions get accumulated in the cell, so that the interaction between positively charged C-terminal domain and the negatively charged membrane is broken. So this acts as a sensor, and when this is broken, there is conformational change such that the pore becomes functional and glycine betaine is brought inside the cell. Now this is a general outline of the different kinds of channels and carriers present in bacterial cells, so that sodium ions are extruded in turn for proton, potassium ion influx and efflux can occur. There are channels for import of compatible solutes. Water can also move inside and outside through the membrane. Some cases they have aquaporin, and then there are some specific channels called MSC. So mainly the osmostress response is regulated through the accumulation of compatible solutes, and meanwhile there has to be a very tight regulation of intracellular potassium and sodium ions. Under hypoosmotic conditions, when the cell has to give out the solutes, it cannot depend on carriers being slower in processing. So there are mechanosensitive channels which basically function because of their interactions with the lipid components of the membrane. Now when there is a change in the osmotic concentration, the cell has to decide which path to take, because stress response is a very energy costly affair. So cell has to choose between proper survival as well as economy. So it can either go for a shortcut quick response where less energy is utilized, or if it cannot withstand that stress with the help of this shortcut mechanism, then it has to spend more energy and adapt measures which can counteract the stress. So in the shortcut part, there are sensors which can sense the stimulus. When the stimulus is perceived, then there is signal transduction and the transporter activity of the sensors gets activated. So there can be solute transport. So this is less energy costly, so cells prefer to go for this way earlier. If the stress cannot be mitigated with this property alone, under those situations, when the stimulus gets generated, it is recognized by the sensor, and then after perceiving, there is a proper signal transduction from one compartment of the cell to the other. There are target molecules which get activated. There may be differential gene expression occurring, and in response to this differential gene expression, the proteins, enzymes, or other parts may be activated so as to respond to the stress. So most of these osmoresponsive elements, they are found embedded in the cell envelope. The first and foremost need for this is because they have to remain at the outer surface so that any change can be perceived. And second, as a part of this, when they come to lie in the membrane, then they are in close contact with the different parts such as lipids or proteins, etc., which are present in the membrane. So when there is any change in the membrane tension, then it can be perceived by the sensors which are lying embedded in the membrane. Now these interactions can be direct, like if there is tension in the membrane, or it may be very specific, that is when there is particular interaction of the protein and some particular lipid present in the membrane. So either by bulk phase or by direct interaction, the signal can be passed on from the membrane to the osmosensors which are lying in part or total within the membranes. So these mechanosensitive channels, three of them are most highly characterized, that is MSCL, S and K. MSCL, that is for large size molecules, S for small size molecules. These are pentameric and heptameric proteins respectively, and they form pore-like conduction channels in the plasma membrane. Now these mechanosensitive channels, they have lipid binding pockets into them. So depending on their conformation, they can bind either with the phospholipids or the lysolipids. So when they are present in one conformation, they are binding with one particular kind of lipid, and as there is some change in the environment which is perceived to the change in the membrane composition, then the conformation changes because the lipid binding pocket has to change accordingly. And when there is this particular conformational change, then their solute transport activities get functional. So when there is sudden osmotic downshift, then the lateral membrane tension increases, which causes conformational changes in the channels, leading to their gating, that is the pore portion, mostly helices, they get rearranged, they get tilted, and all this occurs within milliseconds, so that then the solutes, their massive transport can occur. So